introducing you, if you can get that set up. So next up, we have Simon Kienle from the Steno Diabetes Center in Copenhagen. Simon received their PhD in the Konstanz Research School of Chemical Biology under Martin Scheffner, where they use chemical biology to explore site-specific acetylation by non-canonical amino acids. They established a synthetic biology approach for generation of site-specifically acetylating proteins and studied acetylation in the context of ubiquitin proteasome system in autophagy. And they looked at this at a biochemical and structural level, and they identified acetylated and specific interaction. They performed a short postdoc in research department of Boehringer Ingelheim in 2022 on a continued path in chemical biology to generate chemical tools for modulation of proteins, as well as using mass spec to investigate enzyme activities. Now they're performing a postdoc to investigate precision medicine for identification of biomarker via lipidomics, metabolomics in diabetes and complications in diabetes and neurodegenerative diseases. So looking forward to your talk, which is investigating site-specific acetylation of ubiquitin and the suitability of surrogates for acetylation by genetic code expansion. Simon, take it away.
Yo, I hope you can hear me.
Yeah.
Good. Thanks for the introduction, and also welcome from my side to this great GCE webinar. And I will talk about ubiquitin acetylation and non-hydrolyzable surrogates for acetylation. So as a short reminder, acetylation is a posttranslational modification, and it is installed at the epsilon amino group of lysine residue, and this kind of eliminates the positive charge. This posttranslational modification is installed by lysine acetyltransferases, in short, CATs, and is reversed by lysine deacetylases, in short, KDACs. A prominent example of an KDAC is, for example, HDAC6 or sirtuins, and a prominent example for an acetyltransferase is P300. So now next to ubiquitin and ubiquitin signaling and the cascade, I think I don't need to introduce the ubiquitin cascade again because Katrin already did it quite well. Just a short reminder, ubiquitin is installed to substrate proteins via a cascade consisting of 3 enzymes. And this here is ubiquitin. Ubiquitin contains 7 lysine residues, shown here in blue purple. And ubiquitin is covalently attached to the substrate proteins. And ubiquitin and ubiquitin chains attached to the substrate proteins regulate the fate of the substrate proteins. And this ubiquitin signal is actually converted into specific cellular output, as already Katrin mentioned, by ubiquitin binding proteins. And those ubiquitin binding proteins preferentially bind to the hydrophobic patch around isoleucine 44, which is here shown in orange. So ubiquitin acetylation was described first in 2015 by Otake et al. And they could actually show that ubiquitin acetylation at position 6 and 48 inhibit ubiquitin polyubiquitin chain elongation at several sites. So it is not only ubiquitin acetylated at position 648 does not only inhibit the chain elongation at the respective acetylation sites, which is more kind of obvious since the lysine residue is not available, but it also inhibited chain elongation at other sites, as summarized in this picture here. But besides that, it is actually not much known about ubiquitin acetylation. The only thing besides the inhibition of polyubiquitin chain elongation is that ubiquitin can be acetylated at every single lysine residue within ubiquitin except the lysine residue at position 29. So this is when we stepped into ubiquitin acetylation. And at that time point, neither ubiquitin binding proteins, which binds to acetylated ubiquitin, were known, nor enzymes regulating ubiquitin acetylation were known. So we then used the genetic code expansion to generate mono-acetylated ubiquitin, and I think I also don't need to introduce the genetic code expansion in this audience, since also Catherine quite nicely introduced it also already. But I just want to mention that we kind of incorporated acetylation, in short ACK, or its non-hydrolyzable analogs, 3-fluoroacetyllysine, in short TFACK, or ketolysine, in short CAC, into ubiquitin at every single lysine site. We then performed a biochemical analysis, so meaning that we investigated the auto-ubiquitination activity of certain free ligases with the respective ubiquitin variants, which are depicted here. And what we found is that besides case 6 and case 48, the auto-ubiquitination of the E3 enzyme were repressed when we used lysine, ubiquitin acetylated lysine 11 and 27. So meaning that when we conclude this, we can actually expand the picture from autarkic to a more complex picture where also lysine 11 and 27 contribute to inhibiting ubiquitin chain elongation. Next, we then ask ourselves, what is the underlying reason for that? And first, we thought there may be structural rearrangements. And so we performed protein NMR to resolve the structures of these ubiquitin variants. And what you can see here is a chemical shift perturbation map, in short CSP. And what you need to know to read this chemical perturbation map is actually just the higher the bars are, the stronger is the structural rearrangement of these amino acids at that specific site within this acetylated ubiquitin variant. And this is ubiquitin acetylated at position 63. And from our chemical analysis, we actually know that there was now big influence on the auto-ubiquitination on the E3 enzyme. Thus, we concluded that actually there must be just minor structural rearrangement. And indeed, we found that this is actually looking like wild-type ubiquitin when we acetylate this lysine 63. We then also investigated the other acetylation, acetylated ubiquitin variants. And what we found is that actually all ubiquitin showing a biochemical effect in the auto-ubiquitination show also chemical rearrangements at the hydrophobic patch of ubiquitin, which is around isoleucine 44, and here shown in orange, meaning that this is a strong chemical structural rearrangement at that site. And as I said, this was true for ubiquitin variants which showed strong effects in the ubiquitination assay, in the biochemical assays. And this is also true now for, which is kind of fits quite well to the structure which we identified, because K27 showed a biochemical effect, ubiquitin acetylated at 11, at the position 11, showed a biochemical effect. And also here we found a structural rearrangement at the hydrophobic patch. So we concluded from here that the structural rearrangements are the reason, the underlying reason for the biochemical effects we observed. Moreover, we also found that each acetylation in ubiquitin leads to a distinct structural rearrangement. And first, we asked ourselves, is this biologically relevant? If so, then we would see difference in the binding of ubiquitin binding proteins to those acetylated ubiquitin variants. So we next then performed affinity enrichment experiments. And I think I also don't need to introduce this workflow because Katrin also nicely did that. But just as a short reminder, we used ubiquitin acetylated at the specific lysine residues and incubated this with cell lysate, enriched for binding proteins, and analyzed it via mass spectrometry. As control, we had unmodified ubiquitin. And what we obtained here is this heat map, where each row stands for a specific binding protein, and the color indicates either in red enrichment or in blue depletion. And what we found here is that we had specific binding behavior of binding proteins to each acetylated ubiquitin variant. For example, here with ubiquitin acetylated at position 48. We then also went into detail, in more detail with those binding proteins and confirmed some binding proteins via Western blot analysis. And they showed nicely overlap with the binding behavior which we obtained with mass spectrometry. Just here, for example, NEP52 showed a strong binding to ubiquitin acetylated at position 6 in our Western blot confirmation, as well as in our mass spectrometry-based experiments. And NEP52 actually brought our attention very much, because NEP52 really shows a strong binding to ubiquitin acetylated at position 6. And NEP52 is a super interesting protein because it functions as an adapter protein for ubiquitinated intracellular pathogens like Salmonella. And here, NEP52 normally binds to linear ubiquitin chains by its C-terminal zinc finger. And as I said, then functions as an adapter protein for autophagy, especially xenofraction. So we then wanted to look into more detail with NEP52 and its interaction to ubiquitin acetylated at position 6. And first, we confirmed our results from the affinity enrichment experiments with recombinant NEP52, and could show that also recombinant NEP52 shows a strong binding to K6 acetylated ubiquitin. Next, we also then asked the question, does NEP52 bind to K6 acetylated ubiquitin via its C-terminal zinc finger? And indeed, when we depleted the C-terminal zinc finger, we did not see NEP52 binding to ubiquitin acetylated position 6, whereas when we have the full-length NEP52, we saw binding, meaning that NEP52 binds to ubiquitin via its C-terminal zinc finger when ubiquitin is acetylated at position 6. Interestingly, we could also show that the binding affinity of the full-length NEP52 is somehow comparable to the natural substrate of NEP52, linear dimeric ubiquitin. We then had also a closer look into the crystal structure of NEP52, the C-terminal zinc finger here in green, binding to ubiquitin, and we could see that the lysine 6 here, the acetylated lysine 6 of ubiquitin, points towards a hydrophobic cave of NEP52 C-terminal zinc finger. And so we hypothesized that the acetylation increases the hydrophobicity of the lysine residue and allow a better binding of this lysine residue into this hydrophobic patch, a hydrophobic cave. Next, we also investigated the enzymatic regulation of ubiquitin, and to cut a long story short, we could show that P300 acetylates ubiquitin at position 11 and at position 48, and that HDAC6 acts as a deacetylase for every single ubiquitin acetylated ubiquitin variant. So we can actually add P300 and HDAC6 to the picture of ubiquitin acetylation. We did not only investigate ubiquitin acetylation, we did also investigate NAT8 acetylation. And I think I also don't need to introduce NAT8 as an ubiquitin-like protein, because Katrin did that. What you need to know about NAT8 is that it can also be acetylated at several lysine residues within NAT8. And we again investigated the effect of this acetylation onto the NAT8 cascade. And what we found here is that actually none of the acetylation of NAT8 influences influenced the ubiquitin cascade, except when NAT8 was acetylated at position 11. We then again asked ourselves what is the underlying reason for that and performed structural analysis of NAT8 acetylated at position 11. And again, we could show that this acetylation leads to a strong structural rearrangement in the NAT8 molecule, may be the underlying reason for the observed biochemical effects. So now to the last part of my presentation, which is about the non-hydrolyzable analogs of acetylation. Trifluoroacetylysine and ketolysine have been previously incorporated via genetic code expansion into proteins. For example, in 2010, the group from Wenji Liu actually incorporated ketolysine into proteins and performed oxime ligation. More recently, our group used also ketolysine to perform oxime ligation and to generate mono-ubiquitinated P53. Also, trifluoroacetylysine, in short TFACK, was already incorporated into proteins via the genetic code expansion using the acetyl synthetase system. And here they investigated DNA binding of trifluoroacetylated P53. Besides those amino acids, which structurally nicely mimic acetylation, also often glutamine had been used to mimic acetylation. And this is a work published in 2002 by the group of Wei Gu, where they actually used glutamine to mimic acetylation in P53. And they actually used glutamine because they concluded that also glutamine eliminates the positive charge similar to acetylation. In this respect, they are more or less right, because also glutamine eliminates the positive charge, but there may be different attributes of acetylation which contribute to the effect of acetylation. So we thought about comparing all these amino acids side by side. And thus we incorporated trifluoroacetylysine, ketolysine, acetylysine, and into ubiquitin at position 11, and also used lysine to glutamine or arginine substitution to compare their ability to mimic acetylation. And what you can see here is actually that only the non-hydrolyzable analogs are able to mimic the real acetylation in ubiquitin at that specific site. So only trifluoroacetylysine and ketolysine resemble truly acetylated ubiquitin, whereas glutamine and arginine are somehow different. So we then have a closer look to the amino acids and came up with the hypothesis that maybe the side chain length is one critical contributor to the observed effect. So to investigate that, we then performed again NMR spectroscopy and found indeed that only with the same side chain length, we see similar structural rearrangement in the ubiquitin molecule. Whereas with glutamine, we see a somewhat different structural rearrangement in the ubiquitin molecule, meaning that indeed the side chain length is an important contributor to the observed effect here. And if the structure is really meaningful, we again should see some binding difference of ubiquitin binding proteins to those ubiquitin variants. So we again performed affinity enrichment experiments. In this case, we kind of incorporated these amino acids at the position 11 of ubiquitin. And what we found is that indeed there was only a high correlation of the non-hydrolyzable analogs to acetylated ubiquitin in the binding behavior of the ubiquitin binding proteins, whereas there was only a little correlation of glutamine to really acetylated ubiquitin, meaning that only trifluoroacetylysine and ketolysine can properly mimic acetylation in this context. And then we also thought about to use this in a more in a context that may make most sense. So we went forward to incorporate those amino acids in living cells because there are deacetylase present. So it is most valuable to have non-hydrolyzable analogs. And then we incorporated trifluoroacetylysine and ketolysine together with acetylysine into P53 and H1299 cells. And here you can see the incorporation into P53 at position 120. And what you can see here is that we get nice incorporation of acetylysine with around 1 to 2 millimolar. And this is also true for ketolysine, whereas we got just low incorporation with trifluoroacetylysine. And this was not site-specific. This was also seen with another site in P53 at the site 164. So we also got good incorporation with ketolysine and acetylysine, whereas there barely any incorporation with trifluoroacetylysine. And then we ask ourselves, what about the modification status? We can nicely incorporate acetylysine. Do we actually need non-hydrolyzable analogs for acetylysine? And what we observed here is actually that when we analyze the modification status of these P53 molecules, that the great majority of these P53 molecules are unmodified when we incorporated acetylysine, which makes a lot of sense since deacetylases are present and this is a hydrolyzable modification. It will be deacetylated. However, we also saw that trifluoroacetylysine got deacetylated to some extent, so to a 1-to-1 ratio. Only the ketolysine showed to be completely stable in this setting. So now to the final summary. I hope I could show you that acetylation of ubiquitin influences the structure of ubiquitin and thus regulates the binding of ubiquitin binding proteins and also influences the autoubiquitination of certain E3 enzymes. Moreover, we could add P300 and HDAC6 to the picture of ubiquitin acetylation as acetyltransferase or as deacetylase respectively. And I hope I could also show you that only non-hydrolyzable analogs from acetylysine are properly able to mimic acetylation, and that glutamine is just fully able to mimic all aspects of acetylation. Furthermore, I hope I could show you that the side chain length contributes to the effect of ubiquitin acetylated at the position 11. And in cells, P53 modified with acetylysine or trifluoroacetylysine get fully or partially unmodified, and that in this scenario only the ketolysine is a proper mimic for acetylation and is fully resistant against hydrolysis. At the end of the talk, I want to thank you for your attention, and especially my collaborators of previous lab, like the AG Schäffner lab and Tobias Schneider who did the NMR studies, and the AG Marx for the collaboration. I want to especially thank Elephant Kang and Simon Elsesser and Katrin Lang for sharing the cDNA, which we used throughout the studies. And if you have questions, I'm happy to answer them now.
Thank you very much, Simon. That was excellent. We have a question from Ryan Mell.
Simon, first I want to say to both these speakers, these were both just two fantastic talks on using new technology and also discovering new biology, right, with this new technology. And I really appreciate the thoroughness in which you approach these problems. And my question to you, Simon, specifically is one of the goals of the center is to optimize key technology that just needs to be improved, right? So that more people have access. One of the technologies that has been brought up to us a number of times is acetylation. While encoding acetylation has been around for a while, the non-hydrolyzable mimics are newer. Your demonstration of their utility here is exquisite, right, and fantastic. But one thing that we were confused by in the center was the robustness of these tools for encoding proteins. And we haven't had a whole lot of success in getting the non-hydrolyzable systems to work well, and even acetylation to work well, and having to sort of jack up amino acid concentration and all of that. I would love to hear from your experience, in making all of these proteins and evaluating them. Clearly you're making enough protein to do your studies, but was this a big effort, or were there challenges in how these tools worked, or did you have to make new tools to make this work? How did making all of this material go for you in order to make these studies happen?
Yeah, at the beginning it was quite a challenge, but we then kind of figured out how to do so. You need actually a relative high amount of acetylation to get proper incorporation. So I used actually around 10 millimolar acetylation, which is really high, but it is commercially available, so it was not a huge problem, which is also true for trifluoroacetylation, which is also kind of commercially available and actually more or less cheaper than acetylation. The problem is with keto, you need to synthesize that on your own, and this is kind of a strong disadvantage, and yeah, also limits kind of the application. What I found out is that the new synthetases, which have been provided by Katrin Lang lab, that they work quite well. So they were more stable than the synthetase we previously had. So depending on the system you use, it kind of be working better or worse. So I think I can actually provide the synthetase for that.
Are those synthetases from Katrin Lang's lab available, or is that something that's unpublished right now?
This is, I think they don't have interest in publishing that. There was just kind of a byproduct from the screen that they kind of by accident crumbled through synthetases for acetylation. This was just due. Yeah, they are actually. I talked to them on a conference, and then they offered me that. So I don't know if they have added them to Edgene. I'm not sure because yeah.
No, that's totally fine. That's totally fine. I can always follow up with Katrin about that. I just was curious about how well the tools were working for you, because obviously you've done quite a bit of biology with them. I just didn't know if you were having trouble making material or not. And it sounds like just going to the newer version from Katrin, as well as to high concentrations of amino acid, were sufficient to make your systems work.
Yeah, because this synthetase is often the crucial point within the genetic code expansion. And there was kind of a publication previously that N-terminally truncated synthetases work better. This new synthetase was an N-terminal truncated synthetase, which was easy.
Great. Thank you so much.
Another question from Andy.
Hi, great talk, Simon. Just related to the question from Ryan, the trifluoroacetylene did not incorporate in the cells, eukaryotic cells. Do you think that's a synthetase issue, or what do you think that was?
Yeah, in living cell and cell lines in human cells, it did not really incorporate it well. This is true. I'm not sure if it is kind of an uptake issue or if it is a synthetase problem. For me, it looks more like a synthetase problem, but we never kind of dig into that, and this was really, really low. But we always have at least less incorporation with trifluoroacetylysine when we compare to other acetylated variants. So this is more kind of often seen with trifluoroacetylysine and all of that. So
Thank you.
If there's not any other questions, let's thank the two speakers again. These were just fantastic talks. Thank you.
One more quick question.
Oh, Andy has another question.
All that last stuff says in preparation. When will it be submitted? I'm just so excited to see that come out. And what's your thought about the timing of getting that written up and submitted?
Yeah, we have been working on that for quite a while. I cannot say something because it also then depends. So we did not submit it yet, so this is kind of hard to estimate.
Okay, it's still in preparation.
Yeah, still a little bit in preparation. And in respect to the synthetase, if you may approach, so I don't have them with me now, but if you may approach Martin Schäffner, so the previous lab, the synthetase will be distributed to everyone. So.
Thank you.
Well.
All right, now we can thank the speakers for two fantastic talks. That was just really great. And again, spread the word for bringing in postdocs, senior graduate students to give talks to share their work. I really enjoy having the people doing the actual science give the talks because I feel like you really get to the information. So thanks again, and we'll see you all in a month.
Thanks.
Thanks, everyone.